Okay, looks like we are good to go on YouTube Live. So last week we had a problem with our YouTube Live chat where people weren't able to ask questions. Hopefully that's fixed this week because I have no idea how to fix it.
Same again from my end. Jen.
Really?
Yeah.
Could you see it all right last time, Adam? Or
no?
Okay.
But both of you had it fine, right?
Yeah, which is really weird. And I wasn't logged into. I'm not logged into the cell migration account.
Yeah, I'm in my personal one, but very odd.
That's too bad because we definitely used to get some questions via YouTube, but
let's Google it this week.
Yes, we'll ask the Google. And if anyone watching knows how to fix that, feel free to jump in.
I wasn't logged in. Okay, mine is this week. Yeah, I can't access it either.
All right, well, you guys won't be getting questions via that avenue. We'll have to rely on the folks here in Zoom, but I'm sure they will be super thoughtful and supportive. All right, and we can go ahead and get started in just a minute. We'll have to rely on the folks here in Zoom, but I'm sure they will be super thoughtful. Somebody has the YouTube open.
Sorry, I've just turned it off.
I always end up having to hear echoes of my own voice. Okay, so Madeleine, I don't actually think we received a bio sketch for you. So I'm just gonna go ahead and introduce you just to say that you are an assistant professor at Tufts University in the biomedical engineering department. And today you'll be speaking about cell migration and breast cancer metastasis. So whenever you're ready, go ahead and take it away. Thanks.
Yeah, hi, I'm sorry about that. Hi, everyone. It's really nice to be here. Thanks so much for the invitation to share our work and, to connect with everyone, both all over the world. I actually did my PhD in the UK at King's College London on neuronal cell migration with Pat Doherty, and then moved to MIT to work on cancer metastasis with Frank Gertler, and now have my own lab at Tufts. So I'm going to share some work from that area, and then So my lab is interested in metastasis, and we know cancer affects 1 in 2 men and 1 in 3 women. We focus mostly on triple negative breast cancer, a subtype of breast cancer that has limited treatment options and that is aggressive. And about 30% of patients will metastasize to, defined organs within the body. And the main treatment options still for this subtype, even though there have been a few more that have been approved recently, is still chemotherapy for this particular subtype of breast cancer. And the response rate is extremely variable. And patients, there's a high level of recurrence in patients. And, this really doesn't work very well for patients that already have metastatic disease. And so I won't dwell on this too much for this audience, but, I always just like to remind people that, cell migration is obviously important for metastasis, because still get review comments on some of my grants that ask me why I'm studying migration in the context of cancer. And, both in terms of just supporting tumor growth. So there are, various studies showing that, if tumor cells are able to proliferate and migrate, then they do grow larger tumors. So not only just supporting the growth part of the tumor, but also obviously local invasion and the dissemination of primary tumor cells from other organs to the bodies and to surrounding tissues, reaching the lymphatics and the vasculature. And then also, some really elegant work out of Jacob van Rieden's lab from a few years ago, doing intravital imaging of metastases in secondary organs and showing that tumors that are able to migrate locally and spread out within these organs form larger metastases. And if you inhibit cell motility, you will get less less metastases growing in these organs. So really migration supporting every step of the process of cancer growth and metastasis and dissemination. And so, of course, we know tumor cells are not alone when they're disseminating, and they're interacting with a rich microenvironment composed of stromal cells, the extracellular matrix, vasculature, and nerves. And that, there's a lot of crosstalk between the impact of tumor cells on the environment as they grow, the impact of the environment on tumor cells as they disseminate and activate a range of pathways. And so we're really interested in dissecting this further. And so we asked kind of two main questions in my lab. One, how do cells integrate cues within their environment to locally invade and metastasize? And then how do current treatments for cancer actually impact cell migration and metastasis? And so I'll talk really on the matrix part of my lab, and then Dr. Sam Payne, who's a postdoc in my lab, will talk about kind of the other side of my lab, where we're interested in the electrical properties of tissues and how those impact migration and metastasis. So we know that tumor progression is associated with changes in the matrix that promote invasion. And so there's actually a review that just came out a couple weeks ago from Thomas Cox, summarizing all these nice changes in the matrix that can occur during tumor progression, relating to the composition, the cross-linking and the stiffness properties, other mechanical properties, density, turnover. So there's just a lot of different ways in which these changes can then that happen within the tumor and then can impact cellular properties. And I'll just highlight two focus specifically on breast cancer and the work I've done in the past. And one is the organization of matrix, for example, and with collagen 1, which is the most abundant matrix protein in breast tissue, gets reorganized to these straight aligned fibers during tumor progression, and that this is associated with more invasive disease. And this is work from Patty Keeley's lab originally. And then I did a lot of work during my postdoc looking at the heterogeneous distribution of matrix proteins within tumor tissue, whereby matrix proteins become not only more abundant, so we see here a lot more fibronectin, kind of one of the second most abundant matrix proteins in breast tissue, so more abundant in tumor tissue compared to healthy breasts, but also very heterogeneously distributed in gradients around blood vessels. And, showing that changes in the matrix can not just induce migration, but induce directional movement via haptotaxis towards the vasculature and the edges of the tumor. So just a couple examples to highlight some of this in the context of breast cancer. And so there's been a lot of work in the last 5 or 6 years, really pioneered by Richard Hines' lab at MIT and Alexandra Nava, on characterizing the matrix using proteomics. And so they've really broken down the different categories of proteins that are present, represented in the core matrixome by glycoproteins, collagens, and proteoglycans. And also various studies from the Hines lab and the earlier lab to characterize the composition of breast tissues that contain tumors relative to either a control or comparing tumors of different metastatic potential. And so we have a clear understanding that, there's a lot of matrix proteins that are present in these tumor tissues, much more than just collagen 1 and fibronectin, which, are the most famous and the ones many of us focus on. But there's a lot more there. And so we were really interested in trying to get at, how can we focus in on which ones we want to study and which ones are actually important in this process. And so in general, when you're trying to approach the role of a particular matrix protein in migration, many of these, not all, but some of them are commercially available as recombinant proteins. And, you can study them in vitro. And these assays are great for mechanistic analysis and, defined response to a particular matrix cue. So, we started in this area, and we wanted to really try and understand, which assays are the best to study cell migration responses. And I think this is part of a broader question that my lab is also trying to look at in terms of, how best do we study migration so that it's relevant to cancer metastasis in vivo? And I still think there's a lot of open questions in that area. But we started at least looking specifically at effects of the matrix. And, we know that 3D invasion is, more physiologically relevant to what's happening in vivo. And there's been some work showing that, the spheroid invasion assay accurately replicates the properties that are present in tumors, where, you seed fluorescently labeled tumor cells as like a mini tumor embedded in matrix, and, watch the cells invade and grow over about 5 days to a week. And, this measures migration, but there's also definitely a growth component in there. And so it kind of recapitulates both of these properties that support tumor growth in vitro. But, we know these are more time consuming and require live imaging and a lot of matrix proteins as well to put them in 3D, which is really expensive for many of these that are hard to make and expensive to buy. And so, we were wondering, well, which of the kind of 2 other assays that we commonly use to study cell behaviors are really more representative or predictive of what's going to happen in 3D. And so we looked at 2D cell migration, just plating cells on a substrate and evaluating response in terms of speed and persistence. And then we also looked at cell morphology, which has also obviously been linked to cell motility phenotypes, and tried to understand, which of these 2 assays is more predictive of 3D behaviors when it comes to matrix-driven responses. And so this was a project led by a master's student, Jenny, in my lab, and an undergrad, Anna Weldy. And so we collected data on these different assays in several breast cancer cell lines. And in response to 4 core abundant matrix proteins in breast cancer, and we constructed various predictive models to try and understand the relationship between these assays. So for cell morphology, we just did a 2-hour adhesion assay, looked at cell shape. We know these different proteins, and again, we focused on collagen 1, fibronectin, tenacin C, and collagen 4 for really abundant matrix protein in breast tissue that we could study responses to. And we can see that using the range of the shape parameters from Cell Profiler that these matrix proteins have distinct effects, as we would expect, on the cell irregularity, the size of the cell, and also how elongated it is. So we were happy that we were covering a large space in terms of morphological phenotypes, as shown by the staining here. We then did 2D migration assays, where we just plate the cells on the matrix protein and image them overnight. Again, here we were able to kind of capture a large space of migration phenotypes with distinct effects on both cell speed and on persistence. And then lastly, we looked at 3D spheroid invasion in this model. And again, we could capture interesting differences in how these matrix proteins that were added to our gels could impact invasion and growth in this model. So to put this data together, we did a partial least squares regression model where we take these different responses together, reduce the dimensionality to be able to pull out meaningful relationships between them, and evaluate our model both on its ability to predict unknown proteins in terms of the error, the fit of the model with this R-square value, which evaluates how good your model captures all the data, and then the Q-square, which is the model confidence, which evaluates how confident your model is in the predictions. And so what we found was that actually 2D migration was not really great at predicting 3D invasion in response to matrix, and that the percent error was low for the conditions we tested are unknowns, but the model fit and the model confidence particularly was very low. And so when we tried to apply this then to other proteins, it just wasn't as consistent. And so, but what we found was that the shape parameters, and here we included all shape parameters from Cell Profiler, here the percent error was much lower, and then more importantly, the model fit and the model confidence were much higher. So this really suggested that adhesion is a much better predictor of 3D invasion than 2D migration. And so, that's helped us not only in terms of, when we're screening for new matrix proteins, trying to figure out, which ones might actually be relevant, but also saves us a lot of time on doing more time-consuming assays that we can then use for, other experiments, and just focus on this, very simple adhesion assay to predict the effect of a particular novel matrix protein in 3D. And so, interestingly, this was cell line specific, so we couldn't apply the models between different cell lines. You do need some framework for that particular cell line and the morphology that it has, even within triple negative breast cancer. So you still need some reference parameters for each cell line that you're using to get a confident value. But we were able to predict the effects of novel proteins. And so now, when we try and study a new matrix protein and its effect, we start initially by just looking at the adhesion and the shape. Should I answer? I see there's a question. Should I answer it now or should I wait?
Totally up to you.
Okay, I guess I'll just answer it now. So the 3D matrix concentration, so we used a basis of collagen 1 with a 1 mg per mil. And then we use either 20 to 100 micrograms for the different proteins. So the data are in the paper. So we didn't try a range of concentrations, which is definitely something that would be really valuable to look at as well, because we know some of these proteins have biphasic effects as well. So that's something that would be good to do. So again, these methods are obviously advantageous in many ways to be able to cell shape predictor 2D or 3D. So yeah, so cell shape was able to predict, I guess I'm not sure what the question is.
Well, my question is, when you say cell shape measured in 2D?
In 2D, yeah, measured in an adhesion assay in 2D of just a 2-hour adhesion assay, which for these cells is a good amount of time for them to respond to in a morphological response to that matrix protein, to individual matrix protein, sorry. So just that initial 2D response.
Thanks.
So, studying the response to individual matrix proteins is very helpful, especially in then dissecting more mechanistic analyses of what's happening. But as I showed with some of the proteomics data, there are many, there are over 100 different matrix proteins in our tumor tissues. And so, how do we start to dissect the effects of whole tissue matrix changes on cell migration properties? And so, some tools that people have developed are to use cell-derived matrices. So this is, culturing usually fibroblasts, which do generate a lot of matrix. And these, put them in a dish, let them secrete matrix over about 2 weeks, remove the cells, and then you have a matrix. But the problem is that this is only generated from one cell type. And even though fibroblasts, do generate a lot of matrix, they're by no means the only cells that are contributing. Tumor cells contribute a lot as well, as shown by the proteomic studies from the Heinz lab. And so, you're not really capturing all the matrix that's present. And so we ended up, so another way to then capture more of the matrix is doing tissue decellularization. So taking a tissue, removing its cells using detergents, and then most people end up digesting that to be able to apply in the context of regeneration or tissue engineering or for proteomics. And so obviously when you digest, you lose a lot of the structural information and ECM organization. But we decided to kind of pool these tools together and look at reseeding cells on decellularized matrices. So this work was led by Andrew Wishart, a former technician in my lab, and Sydney Connor, a graduate student. And so we generated breast cancer tumors from both xenograft models with injecting cells in the mammary gland, and then a genetic PYMT mouse model, which is an established mouse model for breast cancer. And we took the tumors out, we decellularized them using detergents to obtain a matrix scaffold. And then we seeded cells on these to be able to look at whole tissue effects on both cell proliferation and cell invasion in these 3D ECM scaffolds. And so we did a lot of characterization, which I won't show here, but is included in the paper, to make sure that we were removing all the cells, that we were maintaining the matrix in terms of composition and also structural properties of the matrix. And we then reseeded cells. What we found was that when you placed cancer cells onto a native healthy mammary gland versus a tumor mammary gland, either from this model or our other model we used, we saw a huge increase in migration. And so this shows that, overall, the matrix changes associated with the presence of a tumor in the mammary gland are pro-invasive, pro-proliferative, that not just all these individual proteins are driving these changes, but that overall the whole tissue changes are increasing this. And this had not really been examined in this way. But from here, we were still, okay, so all these changes together are pro-invasive. So then how do we then try and narrow in on new proteins or, proteins from these about 120 or so that are present that are also contributing to invasion? And so we ended up trying, focusing on another condition, physiological condition that changes the matrix that promotes invasion in mammary tissue, and that is obesity. So obesity affects, 2 in 3 adults in the US, and it leads to a higher incidence of breast cancer, more metastasis, and decreased response to chemotherapy. But more importantly, obesity has been shown to induce fibrosis and change the matrix. It does a lot of other things, as shown here by this diagram, and that's what most studies in the field focus on. And people knew that there was this matrix. It's kind of shown here in the background discreetly, but not as much had been known about what the effects of obesity are on matrix, especially in a tissue like breast, which is high in adipose cells, and therefore very sensitive to changes related to weight and obesity. And so, we were interested in whether the matrix changes associated with obesity and tumor progression were the same, and maybe perhaps that by overlapping, these composition of these matrices, we could identify or kind of narrow in on some core matrix proteins that are important for invasion in this tissue. So we collaborated with a lab at Tufts that had generated obese female mice, that are fed on a high-fat diet for 16 weeks, and again isolated the mammary glands with our decellularization method to get the matrices, and we found that obesity also induced invasion of the tumor cells in this model. And so again, this was the first demonstration that overall changes in the matrix induced by obesity in an adipose tissue like the breast are overall, pro-invasive. So we performed matrix proteomics on the obese lean and obese mammary gland, and then overlapped this with the existing data sets that were out there for tumor matrix composition. And this allowed us to narrow in on a list of about 9 matrix proteins that are upregulated or more abundant in all of these conditions that are associated with more invasiveness. Some of which we knew, again, fibronectin, but some of which had not really been studied at all in the context of breast cancer or effects, detailed effects on migration. So, we tested these in our adhesion assay to see if, to predict the effects and if any of them showed through our pipeline to be, of interest. And the one that we ended up focusing in on is collagen 6. And so collagen 6, induced distinct effects on cell shape in our adhesion assay here. And when we tested it in 3D in our spheroid model as well, we also saw, increased levels of invasion in this model when the cells were incubated with collagen 6. So we turned to then patient samples to take a look to see, can we see collagen 6 in breast tissue? So we can see that there's a high collagen 6 shown here in green. This is a human triple negative breast cancer tumor, kind of at the tumor stroma interface, also abundant in the stroma, and collagen 6 is known to be secreted by adipocytes. And so we were able to show either by protein staining in a larger cohort of patients, or also by mRNA expression, that a lot of patients had at least some level of collagen 6 in their tumors. And that again, using mRNA, which is a huge caveat in terms of matrix, understanding matrix composition in a tumor, but showing that the high abundance of the chains that make up collagen 6 at the mRNA level in patients with triple negative breast cancer had worse outcome than those with lower levels. We then also wanted to confirm whether this link to obesity was also relevant in patients. So we worked with a pathologist at Tufts Medical School to obtain a small cohort of patients with triple negative breast cancer and that had increased BMI. And so we had a healthy group, an overweight group, and an obese group based on the classic BMI numbers. And we saw that within the adipose tissue area, really in the stromal edge of the tumors, the patients that had a higher BMI tended to have higher collagen 6. So this confirmed also a link between obesity and collagen 6 in these patients with triple negative breast cancer. So here's a question on the decellularization of 2D surface. Would osmotic lysis or detergent treatment be a better method to retain the ECMs secreted by the cells? Yeah, so there's definitely a range of detergents that have been used. This method of decellularization has been leveraged a lot by tissue engineers to be able to access matrix for regenerative purposes. And so definitely different methods can lead to different enrichment. And there are a couple studies that compare decellularization by different detergents and show different, that you do get some differences. So I think there's definitely a lot of work to do to optimize that, to, capture more of the matrix or to better understand what you're capturing. And I think one question we often get asked is, does that retain then like the growth factors that get sequestered in the matrix, for example, or, and that's something that we also haven't looked at that is really, would be really critical. So we know we're removing a bunch of cells and we know we're retaining the matrix, but I think there's still a lot of characterization to be done in that area. So collagen 6 is a collagen that is made up of 3 chains, alpha 1, alpha 2, and a third chain that is usually alpha 3, but can be other subtypes depending on the tissue. And interestingly, up until now, there were one or two studies in breast cancer linking collagen 6 to a pro-cancer phenotype, but they were all through this C-terminal fragment called ETP, which gets cleaved by the protein, by proteases, and that this is kind of what was mediating the effects. And so we did a lot of mechanistic work in vitro, which is included in our paper, which I won't show, but to dissect the receptors that drive this response. And what we found was that it was signaling through NG2, proteoglycan CSPG4 receptor, that linked to, that was driving these responses. And NG2 binds to the central portion of collagen 6. And so this also showed that these effects were really independent of this ETP fragment. And NG2 can also signal through, signal also with RTKs. So we tested various RTKs to see which ones were potentially driving this effect. And NG2, we found also signals with EGFR to drive MAP kinase signaling to promote this effect. So we did a lot of mechanistic work in vivo to look at this, but I think that then the value of our system was that we were able to go back into these decellularized ECMs and look at the role of this pathway that we know is activated by collagen 6 in this whole tissue effect. And so, first we use this NG2 blocking antibody in both their lean, obese, and tumor tissue, and found that, if we inhibited this NG2 receptor, we could decrease the migration effect of this decelled ECM on the tumor cells. And so again, this showed that by inhibiting this pathway that is activated by collagen 6, we can reduce the effect of the matrix. And so we did this also targeting other aspects of this pathway, also using matrix from our other tumor. And so, instead of kind of having to go back in vivo and test, which is obviously valuable but way more time consuming, then in this system we can evaluate, does blocking this pathway have any effect on the overall tissue effects on invasion. So another question, does obesity affect mechanical properties of the ECM stiffening? Yes, definitely as well. So we've kind of decoupled that in terms of just focusing on, the signaling aspects of this. But yeah, that's something we're looking into as well and how that correlates. So, but yes. So yeah, so the other part of this I just wanted to highlight is that, I'd mentioned in my postdoc that I did a lot of work on directional migration and, how matrix proteins are heterogeneously distributed in tumors and how that might also impact how they can drive migration. And when we looked at, tumors at a very low magnification from our clinical samples, we can see that there's a lot of the collagen 6 is more on the outside of the tumor. And this is, largely likely because adipocytes, which are more abundant in the stroma around the tumors, secrete most of the collagen 6. But, we could see these endogenous gradients of collagen 6 around the tumor quantified here. And so I was interested in asking whether or not collagen 6 was not just, inducing more migration speed, but also inducing directionality. And so for this, we use this microfluidic device that was originally developed by Jim Baer's lab that I used extensively in my postdoc, where we seed cells in a middle channel, flow in a fluorescent gradient of a matrix protein, and then evaluate directional movement in response to this gradient in a 3D environment. And so we've started working on this. This is still early days, but we do seem to see that when we have a gradient of collagen 6, we can induce somewhat of a directional response in these cells, and that they do seem to orient and elongate towards the gradient. And so this is something that we're pursuing to try and see if collagen 6 is also not just, again, inducing more speed, but inducing directionality as well. And promoting, attracting these cells into the stroma, which then obviously takes them into, closer to be able to invade into the vasculature and disseminate throughout the body as well through that, or the lymphatics. So to summarize, so far we've developed or optimized this novel method to study whole tissue ECM effects on cell behavior independently of what cell type secreted it. So we're just taking what's there and looking at the effects. And we see that obesity induces changes in the mammary gland ECM that overlap with the ECM of breast tumors. And collagen 6 is one matrix protein that we focused on, which is upregulated in both of these contexts that we validated in patient samples, and that can induce breast cancer cell migration and also seems to be driving directionality as well. And that if we block the signaling pathways that are activated by collagen 6, we can reduce the effects of the whole tissue ECM. And so, we want to try and see if, targeting some of these pathways then in vivo can also provide novel strategies, particularly for patients that are obese, which have a high abundance of this protein being secreted by their adipocytes. And so to finish, I'll just give a little a snippet of something unpublished that we're also just thinking of, just to bring this, as we think also about cancer, we need to think about treatment. And so most patients with triple negative breast cancer, will get a diagnosis and will immediately receive neoadjuvant chemotherapy treatment. So before even undergoing surgery. And the two most common drugs are paclitaxel and doxorubicin. Also, some will receive cyclophosphamide and some cisplatin. And this, is the goal is to kill right away what might be present, prevent any dissemination early, and then patients will undergo surgery. But about 25 to 30% of patients will exhibit recurrence within 5 years. And so, this asks us well, what is chemotherapy doing? Is chemotherapy, yes, it's killing cells, but what is it doing to to the other aspects of this tumor. And as a reminder, all chemotherapy drugs target cell proliferation, right, which is distinct from cell migration. And what are these drugs doing? And so interestingly, there are a couple studies that have done RNA sequencing of residual triple negative breast cancer cells in patients, and done gene enrichment analysis on these cells. And what do they find? Well, tumors that have high cell migration or low proliferation tend to be more resistant to chemotherapy. And interestingly, genes associated with matrix remodeling were upregulated. And we know that chemotherapy induces huge amount of inflammation in the tissue, which recruits a bunch of stromal cells, all of which can secrete matrix. So we've been interested in trying to understand, how does chemotherapy impact migration? How does chemotherapy impact the matrix and the matrix effect on migration? So in my postdoc, I did start doing some work on this, on trying to evaluate how does chemotherapy drugs impact migration versus tumor size versus metastasis, because effects on drugs in patients is always measured by tumor size. That's it, right, clinically. But again, migration we know is not captured by that. And so what's happening? So I did some basic studies where we would treat human breast cancer xenografts with chemotherapy regimens that are similar to those in vivo used in patients. And so Taxol we know stabilizes microtubules to prevent cell division, and doxorubicin intercalates in the DNA to cause DNA repair, DNA damage, and prevents the repair and leads to cell death. And so, when I would use these with regimens that were published, we could see that we would stop the tumor size. So if you look at the relative change in tumor volume, control mice at this time point keep growing, whereas here these mice stop growing. These are with relatively large tumors that were already metastatic, but that was the clinical setting we were looking at. But when you measure tumor size, these drugs are doing what they're supposed to do. But then I also did endpoint intravital imaging. And I looked at, these tumors, these exact tumors that were shrinking, how was cell movement inside these tumors right before sacrifice? And interestingly, the effects on cell migration are very distinct than the effects on tumor size. So the Taxol treated tumors actually did have a decrease in cell migration in vivo. Again, this is by intravital imaging. But the doxorubicin treated tumors did not, And so this kind of suggests that, yes, again, these drugs are killing proliferating cells, but we need to also be mindful of what they're doing to migrating cells that might leave behind these cells that are highly motile, which is obviously not what we want for these patients. And that we've also repeated this from the matrix side to be like, well, what's happening in the matrix? So this is work led by student Jackson and Justine in my lab. Different mouse, this genetic mouse model, again treated with these drugs, shrink tumor size, no effect on metastasis. What are they doing to the matrix? Well, what we found was that when we used our reseeding method to isolate the matrix from these chemotherapy-treated tumors, they did increase migration of our tumor cells. So this suggests that this chemotherapy-treated matrix is pro-invasive, pro-migratory. And so, we did proteomics on this matrix, and we can identify, again, candidates. And here's collagen 6, which is the one protein I talked about, which we also found upregulated in paclitaxel treated tumors. And so, we're currently investigating this and, doing some staining to validate that these proteins are in fact more abundant, and looking at the signaling. We have a couple other candidates that we're working on. And so, all this to say that, chemotherapy, which is, our standard of care for all metastatic diseases that have a high abundance of migratory cells, do a great job of killing cells, but are they also doing things within the tumor that could be promoting invasion? And one way is by not necessarily inhibiting migration of highly migratory cells, or also by inducing so much inflammation that you're actually generating a pro-invasive ECM environment. And so that, understanding how these treatments actually impact cell migration and how that relates to metastasis is really critical if we want to tackle metastatic disease, which, as we know, is refractory to many treatments, and that once the disease is disseminated, it's really hard to treat. So just something to keep in mind in terms of studying migration in cancer. So with that, I will thank the members of my lab I highlighted throughout the talk. And here we are. The students that did the individual work, our collaborators that I highlighted as well, and our sources of funding. And thank you for your attention. And I see there's a couple more questions, so I can take those, I guess.
We can help you out, Madeleine, by asking them for you and sifting through the chat. So we'll just do those quickly. So back in the beginning, Ernest Liu asked if cell shape and cell 2D migration are correlated, why is 2D migration not predictive of 3D invasion?
Yeah, we found that it was more like the persistence was more predictive than the speed. So that's really interesting. And then, yeah, I saw after actually that came in after about a matter of directionality and persistence. So I think, here we were looking at completely random migration. So obviously there's no directional cue. So, where that persistence is driving, I think that's a really important question and aspect to look at. So yeah, how does their directionality of the cells without a directional cue, right? Just how persistently they migrate. I think that's something we want to delve in further.
Thanks. I think we had a question from Mohammed, said it might be interesting to observe collagen 6 secretion and metastasis correlation with lipoma, since adipose tissue seems to be secreting collagen 6.
Yeah, no, we're definitely, that's a lot of work that we're doing now is on that side is trying to understand how obesity induces those changes in the breast tissue. So yeah, we're looking into that more closely and in that relationship. Yeah.
Great. Okay, 2 more quick ones from Agata. Was the collagen 6 included in your initial study, and then was the response to it different than from the other ECM proteins?
So we did not include it, but we tested it after as a validation for that. And so we did see that it was really well predicted. So that was good, but that wasn't part of our initial study. Yeah.
Great.
Last one from Zoom. Vicky Sanz Moreno asks, have you looked to immune cell infiltration after chemo?
Yeah. So that's kind of our next thing is so who is secreting this matrix and who's coming in that could be doing that. So yeah, we are looking into that, and that's kind of the next step after hopefully we finish this and get that published. So yeah, we'll see.
Thank you so much. All right, Sam, if you want to go ahead and start sharing your slides. And Madeleine, I just wanted to say, I think it's really, I love that you guys are looking at the migration effects of these commonly used chemo drugs. That's really, really important. And now Adam is going to introduce Sam for the next part of our talk. Thanks, Madeleine.
Thanks, Madeleine. So our second speaker is Sam Payne. Sam did a PhD in chemical engineering and applied chemistry from the University